Greetings, students. This is a recording for Biology 1406. It's part one of the lesson on cellular respiration. So cellular respiration, as you all know, is the process by which cells extract energy from organic molecules to regenerate their supply of ATP, which is continuously being consumed. And so there must be a continuous process of regenerating that ATP, and that is the main business of cellular respiration. All right, so when we say that cells extract energy from food to generate ATP, that's what we mean when we're talking about cellular respiration. And food can be any energy-rich organic molecule that has carbon and hydrogen in it. So that's going to be carbohydrates, pretty obviously, but also lipids, oils, fats, the fatty acids particularly. Even glycerol can be used as a food in the cellular respiration pathway, and of course amino acids can be used. So any organic molecule can be used as food or fuel to generate ATP. And of course that process is catabolic in that the large organic molecule is broken down into its inorganic components. And that process of taking a complex molecule and reducing it to simpler pieces is referred to as catabolic metabolism. All right, so we're going to take a look at the pathway, the central pathway of cellular respiration, and then at the end of the chapter on the subject, we kind of take a look at some related material. The key, as this statement here says, a key thing to pay attention to when we look at cellular respiration and photosynthesis is the movement of energy via the movement of electrons and hydrogen atoms, the proton plus the electron around it, as we oxidize or reduce various molecules. Catabolic metabolism, like cellular respiration, is an oxidizing pathway. Photosynthesis involves a reduction pathway where we're adding electrons and protons to a molecule to make it larger and more complex. All right. So redox, we're just as a combined statement that means reduction and oxidation reactions. And photosynthesis and cellular respiration both involve reduction and oxidizing processes, although if you want to characterize them overall, cellular respiration is primarily about the oxidation of fuel, and cellular respiration is primarily about the reduction of carbon dioxide by adding hydrogens to it to make sugars, HCO, sugars. But so in all cellular respiration and photosynthetic pathways, we've got something that is being oxidized and something that's being reduced. So in cellular respiration, the fuel is the agent that is being reduced, the reducing agent. The oxidizer is oxygen. And the result is oxygen has been oxidized by adding electrons and hydrogens to it to make water, and the fuel has been reduced by stripping electrons and protons off of it, turning sugar into carbon dioxide. So water and carbon dioxide are the products of a reaction between sugar or any other organic molecule, but preferentially sugar, and oxygen. All right. A key actor in the pathways for cellular respiration, as well as photosynthesis when we get there, are electron carrier molecules, like this one right here, NADH. We'll also see that we have FADH, and we have NADPH in photosynthesis. But NADH and FADH are participants in the cellular respiration pathways. And what these are, you might recognize this. There's a phosphate group right there. There's a pentose sugar. It's got 1, 2, 3, 4, 5 carbons, and a nitrogenous base. In this particular case, this nitrogenous base is a purine. And then we've got another one of those molecules over here where the nitrogenous base is a pyrimidine. So these are very closely related to, and in fact are modified versions of the same molecules that we build DNA and RNA out of. They're closely related. ATP is a modified nucleotide, as in DNA and RNA. All right. Yeah, and here we go. Here is NAD with a plus on it. This is the oxidized form of this molecule. It's missing electrons. It's missing some hydrogen ions. And here's NADH, the reduced version of this molecule. All right. And notice we've added another hydrogen in this location right here, another proton. And so there's got to be electrons making that bond right there. So we've added another atom in here. We've gone from a slightly simpler molecule to a slightly more complex molecule here. And this is a means of storing and moving electrons and protons from one place to another via the conversion of this molecule into this form right here. All right. So the role of ATP, which is another modified nucleic acid. Here's a purine nitrogenous base. Here's a 5-carbon, 1, 2, 3, 4, 5-carbon pentose sugar, ribose, and then 3 phosphates in a row. So this is adenosine triphosphate. If it only had 2 phosphates on it, that would be diphosphate. If it only had 1 phosphate on it, it would be monophosphate. And all 3 of those things exist. Here we've got ATP being converted into ADP plus a free phosphate ion. That would be this just hanging off by itself in the cytoplasm of a cell. All right. So that process of gaining or losing a phosphate group is called dephosphorylation. And classically what happens when ATP powers a chemical reaction somewhere, what it really is doing is the ATP is transferring its 3rd phosphate. Sometimes ADP is transferring its 2nd phosphate, but classically it's ATP, transferring a phosphate to a protein, thus energizing it and giving it the power, if you will, to catalyze some chemical reaction. So for instance, we looked previously in the previous unit about, we looked at proteins that acted as pumps moving things across the cell membrane, and those pumps required energy, and some of them were powered directly by ATP, and that's what would happen. Add a phosphate group and that protein changes shape, it can power the movement of things against their concentration gradient, or power the synthesis of something by putting 2 things together. The phosphate group represents that transferred energy. All right, we all know, hopefully, that the mitochondrion is a central key organelle in cellular respiration processes. All right. So in the mitochondrion primarily, but also in the cytoplasm of a cell, we have a process where an ADP, product of this reaction, ADP right here, is regenerated by having a phosphate added to it, reforming ATP. Now this requires energy. Taking 2 small things and putting them together into a larger, more complex molecule requires additional energy to reattach that phosphate and electrons to couple them. All right, and so that process is driven. It is driven by some other process that gives off energy. So this is an energy-consuming reaction right here, ADP plus phosphate to make ATP. To regenerate ATP, to recharge that battery, requires energy. So that must come from some energy-releasing process right here, where we're stripping energy out of an organic molecule like glucose. Okay, and there are 2 ways in which ATP is regenerated in the cellular respiration pathway. There's one that's called substrate-level phosphorylation, and that's where some enzyme basically does this process in reverse. The arrow would be facing this way. This enzyme transfers a phosphate to ADP, regenerating ATP, and then that enzyme is recharged in some other process somewhere. That's substrate level phosphorylation, this process running in this direction. Chemiosmosis is another way in which ATP is regenerated in cellular respiration, and that is unique to the mitochondrion and to those single-celled bacteria, well, those prokaryotic bacterial cells of which the mitochondria is a distant relative of. Those cells have the ability to do this chemiosmotic process where basically you're mechanically regenerating ATP using a fairly interesting structure that we'll look at later called ATP synthase, ATP making enzyme. And as you can see right here, that chemiosmotic process is the main way in which we make ATP inside cells. All right, you do need to know this summary reaction right here for cellular respiration. This is a glucose molecule or a fructose molecule. They're both 6-carbon sugars, but glucose, galactose, fructose, all the same formula. That sugar plus oxygen, and for the balanced reaction, 1 sugar molecule plus 6 oxygen molecules yields 6 CO2 molecules plus 6 water molecules, and about, it varies a little bit, 36 ATP is stored energy. That's the summary reaction for cellular respiration. But in fact, there's a terrific number of steps along the way to get these reactants to generate these products. There's quite a bit of it in here. And we'll take that process, that whole series of steps, and break it down into 3 major bits. So there are 3 big bits that you are required to know in addition to this summary reaction right here. So memorize this thing, and then we'll apply it to this. Glycolysis, splitting glucose. Lysis means to split, and glyco refers to glycosides, glucose. Oxidation of pyruvate in the citric acid cycle. This is where we take what's produced in glycolysis, run it through another series of reactions. The product of glycolysis is pyruvate, or one of them anyway, a waste product of that. And we take apart that pyruvate molecule in a series of reactions called the citric acid cycle. We store a whole lot of electrons there. We store a few electrons in this process. We make a little bit of ATP here. We make a little bit of ATP here. And then all the electrons that we harvested that have been transferred to NADH or FADH, that's the names of those molecules when they've got the electrons attached to them, the reduced state. The electrons we strip off of strip off of sugar in glycolysis, the electrons we strip off of pyruvate in the citric acid cycle, all those electrons are passed into a process that leads to oxidative phosphorylation, which is where the bulk of this ATP is generated. Okay, so glycolysis, splitting of sugar. What I need y'all to know, all right, is what the inputs of that process are and what the outputs of that process are. So what goes in is glucose, something to accept electrons, that's NAD. This is the oxidized form of this molecule. It's waiting to accept electrons and another hydrogen ion right here. We're going to need some ATP, and we're also going to need some ADP that needs to be recharged. And the end product is 2 pyruvates. These are these molecules right here, the 2 halves of a sugar molecule. We've added protons and electrons from the glucose to NAD, to both of them, making NADH, so these guys are storing more energy now. We've also generated 4 ATP, but we spent 2, so we have a net of 2 ATP. The net is 2 new ATP. We spent 2 ATP, but then we regenerated 4 ATP, so we made 2 net ATP. We've got 4 in total, but we spent 2. Those are the details. Please know that. That's the minimum that you've got to know. Now here we look at it in a bit more detail. The things that I'm not going to ask you to know, but I'm showing you, just to give it a little bit of life to it, is the names for the intermediates as we split glucose apart to finally make pyruvate. We're not here yet. This is just the first part of the reaction. So we take sugar, We crack it open, we modify it, we use some ATP to power this process. So notice ATP right here and ATP again right here. That's where these phosphates are coming from. These phosphates, this is from the first ATP. This one over here is from the second ATP. And we use that to pay for the process of splitting the glucose molecule apart into two isomers, 3-carbon sugars right here, dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. And then this thing gets converted into glyceraldehyde 3-phosphate by an enzyme right here. So there's a little step right here. That's the first 5 steps. All right. There's an enzyme that catalyzes every dang one of these reactions. Then the second half of glycolysis, steps 6, 7, 8, 9 through 10, is the modification of glyceraldehyde 3-phosphate into its final form of pyruvate right here, and that's where we extract some energy from it to regenerate an ATP. Then we do it again right here to regenerate another ATP, and then we do that for the other half. I'll go back here. Remember, there were 2 of these, 2 3-carbon fragments of our original glucose. So that's something that gets a lot of students right here. Each of those runs through this pathway right here. So one half gets you 2 ATP. The other half of the glucose running through here gets you 2 more ATP for a total of 4 ATP. But remember, we spent 2 right here, so that's why it's a net production of 2 ATP right here in total. You'll have to go back over this a few times. And pyruvate is the process here. Every living cell on Earth does this one way or another and generates 2 ATP from a glucose molecule or something similar. All living cells do that. All of your cells do that. Now, in the world of eukaryotic organisms, only those organisms that have mitochondria can go on to the next portion of the process, which is the oxidation of pyruvate and the citric acid cycle. That requires a mitochondrion. All right. Here we're looking at the whole thing put together. And remember, this runs through the second half of the reaction, and then this thing runs through the second half of the reaction, both halves of the original glucose. This animation right here, we'll see if we can make it play. All right, we're going to go ahead and play this video and see how this looks. I might stop and interrupt it here and there, but let's see. I just wanted to highlight this in case you didn't recognize it right here. This is that NADH nicotinide anhydrous dinucleotide. The D for dinucleotide. So this is one nucleotide. That's a phosphate. This is your ribose sugar, 1, 2, 3, 4, 5 carbons, and this is a nitrogenous base, which is a pyrimidine, 1 carbon ring. And then over here, a phosphate, another 5 carbon sugar, ribose, and a 2-ring nitrogenous base, a purine, and that's the NADH molecule. All right, so that was hopefully a useful video summarizing everything we've talked about so far. All right, so next, pyruvate for those cells that do have mitochondria. Pyruvate's going to go through a series of reactions, starting with the oxidation or transformative reaction. And let me show you something. I'm going to run to the very end of this process right here. Let's see here. Yeah, so in the absence of oxygen, metabolism without oxygen, and this is also true for cells that do not have mitochondria, including a lot of anaerobic bacteria, glycolysis still yields those 2 ATP, and then they have to do something else, all right? And those two processes, the two that we ask you to look at, there's some others, are lactic acid fermentation, which is what occurs in the absence of oxygen in an organism like you. When you overuse your skeletal muscle cells, there's not enough oxygen getting there, so your mitochondria can't work with the products of glycolysis. They can't work with pyruvate, and so the pyruvate has to be gotten rid of in a process called lactic acid fermentation. For single-celled organisms living in water, the same process is accomplished more efficiently by alcohol fermentation. And then, of course, we take advantage of this process and use fermenting bacteria and eukaryotic cells like yeast to produce alcohol for beverages, or CO2 and alcohol for leavened breads and things like that. When we cook them, the alcohol bakes off, and the CO2 is really what we're after, because that makes the bread rise, and various other things like that. So that's a different pathway, and that's what's done in cells when they don't have oxygen and or don't have mitochondria. Roughly the same thing. We have to have one of these two pathways or some other fermentation pathway to get rid of the pyruvate. All right, but we're going to go back and look at what happens when we do have oxygen and mitochondria present, and that is the oxidation of pyruvate. All right, so those two pyruvate molecules are pulled into the mitochondria, transported into a mitochondria, and each one of those is going to be converted into acetyl-CoA. And acetyl-CoA is a 2-carbon sugar, the acetal molecule, and then there's a big enzyme attached to it. That's where this cofactor A or coenzyme A comes from. But the acetyl molecule is a 2-carbon sugar. So that means the other carbon has to go somewhere, and where it goes is CO2. So here's the first of the CO2s that you breathe out, and where they're coming from. This is one of the sources of that CO2. And what we did is we took this 3-carbon molecule and we split off a CO2 and left it as a 2-carbon molecule, and the electrons that were holding this together, we stripped those electrons off from pyruvate and introduced those electrons onto NAD, thus making it into NADH. The electrons are here now holding this extra proton on this molecule here, and the CO2 is released as a byproduct, as a gas, because we broke the bond holding that CO2 on. We stripped these electrons out and put them on this molecule, and so the CO2 was released. We'll do something with these guys later on. Right now we're going to follow this. So what's left right here, the acetyl-CoA molecule, is now going to be transferred into the mitochondrion for further work. So the inputs, 2 pyruvates go in. yield 2 Na and 2 NADs must go in. 2 coenzyme A molecules go in. The products are 2 CO2 molecules, one from each pyruvate. This once, this twice. 2 NADHs, this once, this a second time for the other half of glucose. And 2 acetyl-CoA molecules, these guys, which we'll do more with. All right, the citric acid cycle, all you need to do is know what goes in and what comes out. So this is large and complicated. We could require you to know the name of each one of these intermediates right here, the names of each of these intermediates, the enzymes that catalyze all these reactions, and if indeed you go on and take cell biology, the advanced course, you'll have to learn this stuff, but right now it's not necessary. The key thing that's going on in this citric acid cycle is that we've got a molecule. It happens to be called oxaloacetate, which is a 4-carbon molecule. It's very low on hydrogens, rich on oxygen right here. So it's like a really, really bad sugar, a poor sugar. And we're going to attach the acetyl-CoA molecule to that. So we make a bigger 6-carbon molecule that's got quite a few more hydrogens attached to it. And then we're going to manipulate it a few times to strip those hydrogens off and transfer those hydrogens to an electron carrier. Sorry, strip off those electrons and their hydrogens and transfer those to an electron carrier. So NAD is now NADH plus. NAD plus is now NADH. And since we stripped some electrons off of here, we broke a bond, and a carbon dioxide is released here. And then we repeat the process. Another carbon dioxide is released right here. We generate some ATP, or in some cells, some GTP. Same thing. Similar thing, I should say. Then we generate another molecule. We get some more electrons off and take FAD, another dinucleotide, and we add some electrons and protons to it. It's been reduced, FADH2 now. Then we do another NAD right here. So there's a whole bunch of steps, and here we've stripped off the electrons, and here we're regenerating the oxaloacetate so it can accept another acetyl-CoA, and we repeat this process, and that's why it's called a cycle, because all of these things are involved in a cycle that keeps regenerating oxaloacetate so that we can add acetyl-CoA to it to make a new molecule called citrate. That's why we call it the citric acid cycle. And then we go through a long process of stripping the excess energy of acetyl-CoA off in here, getting back to acetyl oxalate, and repeating the process. So we've got a summary reaction right here. Citrate is oxidized. We make 3 NADHs, 1, 2, 3, 1 FADH right here, 1 GTP or ATP right here. And that's for 1 acetyl-CoA. So we're going to run another acetyl-CoA through this process as well, 2 of them to account for the entire glucose. So the net reaction for the entire glucose, if we have the citric acid cycle next, is that we'll make 6 NADHs, 2 FADHs, we'll release 4 CO2s, and we'll make 2 ATP or GTPs right here, guanosine triphosphate or adenosine triphosphate. And that's the process. Taking the original glucose, running through these processes, running through the oxidation of pyruvate. Each of those once will give us 2 carbon dioxide. Each of those halves of the molecule running through this process right here will give us 4 more CO2, 2 for one half, 2 for the other half. If we add all of that up, it's 6 CO2. So our sugar is gone. Glucose is gone. It's been destroyed. All the carbons and oxygens have been released as CO2. And the electrons and protons and hydrogens are all captured either as NADH or as FADH in all of these steps. And that's the citric acid cycle. And here it is in a little more clarity. So again, NADH once, twice, thrice, FADH once, GTP or ATP once, for 1 acetyl-CoA, and of course 2 of these run through this process when we're accounting for an entire glucose. So here we kind of summarize it. So far, for glucose, we've got 4 ATP, 2 from glycolysis, 2 from the citric acid cycle. We've got 6 CO2, 2 of them from the oxidation of pyruvate into acetyl-CoA, and 4 from the 2 runs through the citric acid cycle for each of these pyruvates, or each of the acetyl-CoAs running through this. And we've generated a total of 10 NADHs, 2 from glycolysis, 2 from the oxidation of pyruvate, and 6 from the citric acid cycle, and 2 FADHs from the citric acid cycle. Glucose is gone. All the electrons that we can harvest have been harvested, and indeed we got a little bit of ATP while we're at it. But what do we do with all those electrons? Where is most of the energy? Y'all think about it. Are they in ATP, CO2, or in these electrons being carried by NADH and FADH2? Well, the answer is oxidative phosphorylation, a process occurring at the end of this sequence, if you will. All right, and as it says right here, most of the ATP that you produce is done here. It's a mitochondrial reaction. It's the one place where we actually need O2. So if we didn't have these mitochondria and didn't need all this ATP produced this way, we could do without oxygen. But we need all the ATP. We have mitochondria that make lots of ATP in the presence of oxygen, so we need oxygen. And what happens is, in this process right here, the energy of these electrons is lost as the electrons are allowed to basically drop downhill, losing their potential energy, high potential energy, low potential energy. And as they give off their energy, they do some work. They pump hydrogen ions, and then those hydrogen ions create an electrochemical gradient, which is enough to power the process of making ATP. So here's the key bit. This is called the electron transport chain. And each of these is a protein, or a complex structural protein right here. And each one of these is a little more electronegative than its predecessor. NADH is not very electronegative. This first protein's a bit more electronegative, so it tends to yank electrons off of NADH and take them. Kind of like chloride does to sodium when we get sodium chloride ions. The chloride ion is much more electronegative than the sodium, and so the chloride ion can take an electron right off of sodium, and then the two attach to each other because of charge differences. So it's kind of the same thing going on here. So the electron's stripped off of NADH, we're back to NAD, and a proton floating around here. And then that electron is in turn stripped off of this and pulled to this right here, because this Q protein here is even more electronegative than this one. All right? And in turn, the electron's stripped and pulled over to this proton, to this big protein, which is even more electronegative. It, in turn, has its electrons taken by cytochrome C right here, yet another protein, which in turn, this takes the electrons from, pulling them off even further right here. So this is the electron transport chain. This is the most electronegative. NADH itself, but certainly this protein, is the least electronegative. So electrons fall in this direction. And as they do so, they power a proton pump, yet another proton pump, and a third proton pump. And as you can see, NADH releases electrons that flow through all of this. So each electron pair, there's 2 that go through here, pumps one proton, and another proton. FADH is not quite so, it's a little more electronegative, so it can't hand off its electron here. It's actually more electronegative than this, but it's less electronegative than this protein here, than this protein here. So FADH transfers its proteins here, which run through this proton pump and this proton pump. So FAD, the 2 electrons coming off of it, pump 2 protons. And the protons are storing energy here. All right, we'll go ahead and take a look at, let's see if I can get this to play right here. No, I can't do it. I'm going to pause this. All right, we're going to watch this little animation here. All right, just to get a couple of more things in here. So what we're looking at is the outer mitochondrial membrane, kind of in blue and gray up here. They don't have it highlighted very well, but that's the outer membrane of the mitochondria, what you would see on the outside of the mitochondrion. This is a space between the outer membrane and the inner membrane. This is the two layers of phospholipids facing each other, tails in the center here, heads on either side. So this is the phospholipid bilayer. There's another phospholipid bilayer way out here, and this intramembranous space is where all these protons are pumped. So what do you think? If I have a liquid here that's full of hydrogen ions, what would I call that liquid? There's a question for y'all. And so this electron transport chain, as I said before, strips electrons off of NADH here, or FADH right here, and those electrons are transferred down the chain, each one being more electronegative until we get to this one right here, where we use oxygen, which is even more electronegative than all of these guys, to pull the electrons off along with protons and form water molecules. So the purpose of oxygen is a place to dump these electrons, because I can't keep having electrons pass through here if they pile up right here. They've got to go somewhere. So they bind with oxygen to make water, and then the water dissolves out via osmosis in every which direction and this process can keep running. There are toxins and poisons which are extremely deadly, including cyanide, that interfere with this process right here. So cyanide is a deadly toxin. What it does is it blocks cytochrome C right here so that it can't accept electrons. It blocks the electron accepting site. And so the electrons pile up here, pile up here, pile up here. No more electrons can be accepted by the electron transport chain, so we go backwards. The citric acid cycle can no longer work because NADH and FADH aren't turned back into NAD and FAD here, so they're not over there to pick up more electrons. So everything grinds to a halt and you die. It's extraordinarily toxic because it blocks this guy right here. But when this system is working correctly, we dump our electrons right here, with oxygen being the final electron acceptor, and then this high concentration of protons right here passes back down from high concentration to low concentration. And again, I want to ask you, what do we call this liquid here with lots of hydrogen ions in it? But it passes down its concentration gradient here through this machine right here, which generates ATP. It puts ADP and phosphates together to make ATP. So it constantly regenerates those. And the reason the protons fall back down through here is because there's a lot of protons out here, a lot of hydrogen ions, proton, hydrogen ion, same thing, and very few of them here. Additionally, hydrogen ions are positively charged, and the inside of every living cell is negatively charged. So they're really pulled like magnets down through here, as well as pushing down here due to diffusion. All right, we'll go back to our PowerPoint and finish that off. So oxygen is the final acceptor of these electrons, and when we add an electron to oxygen and a proton to oxygen, we have reduced it and formed water right here. So we make water in our mitochondria, and we use up O2 in the process. And we strip the electrons off of NADH, and we strip the electrons off of FADH, regenerating NAD and FAD, which can go back and be used in the citric acid cycle and even all the way back in glycolysis in the case of NADH to allow those processes to continue. Again, I'm not going to ask you to remember the names of all of these enzymes, the detailed names of all of these structures. That would be nice, but it's not required. I won't test you on it. But I do want you to know what goes in and what comes out. So what goes in is NADH and FADH. What goes in is oxygen. All right. What goes in is ADP. What comes out is water. What comes out is FAD. What comes out is NAD. And what comes out is ATP, shown at the end. It's actually, it'll be the next slide. But those are the total products of this electron transport process. All right. And of course, the last thing right here, so an input is ATP plus phosphate, and output is ATP, is the ATP synthase complex that hydrogen ions drop down through to generate ATP. So in all of this process, the electron transport chain plus chemiosmosis, the power source to actually make ATP is not glucose, it's not NADH, it's not FADH, it's not even electrons. Technically, the power source that literally makes these ATP right here is hydrogen ions falling down their concentration gradient. Now, they got here due to the work of electrons, which were brought here by NADH and FADH. Where did those electrons come from? They were stripped off of sugar in these reactions right here. We generated NADH. We generated FADH right here. They were even produced way back here. We generated some NADH right here in the oxidation of pyruvate. And of course, all the way back here where we generated some NADH right here in glycolysis. So all of that stuff ultimately feeds into this process, generating a power gradient which pumps protons, and then those protons power the ATP synthase mechanism. May seem complicated. It's true, it is complicated, but that indeed is how it works. There's the outer mitochondrial space, here's the inner mitochondrial space, and there's not just going to be one of these things. There's thousands and thousands of all of these things plugged in all over the place inside the mitochondria here. Lots and lots and lots of copies of the enzymes that run the citric acid cycle and all of that sort of stuff. And don't forget, glycolysis is occurring out here in the cytoplasm of the cell, in the cytoplasm. And then pyruvate is converted into acetyl-CoA as it passes through this membrane right here, transferred into this inner space right here where it runs through the citric acid cycle. All right, and if we add all of that together, it varies a little bit, but somewhere between 30 and 36 ATP per glucose molecule. All right, and that varies a little bit right here. And as it says right here, and these are kind of handy numbers, they're real similar to each other, all of those ATP molecules represent about 34% of the energy that's stored in one glucose, 34% of the energy. So my question to you is, where's the rest of that energy? Where's the other 66%? Where does it go? Where is it? Because the first law of thermodynamics says that we can change energy from one form to another, but it's neither created nor destroyed. Think about the second law. That's part of the answer. All right, we talked about this, metabolism without oxygen, and we'll come back and look at it one more time real quickly, but let me go ahead and show you this little video right here. All right, so that animation, hope was useful. And when they refer to the matrix, that's this inner space inside the inner membrane. There's an outer membrane, an inner membrane, and then that space inside where the citric acid cycle reactions occur and where ATPs are generated is called the mitochondrial matrix. It's just the inside of that inner compartment. All right, so we want to talk a little bit about lactic acid fermentation and alcohol fermentation. All right, this is the more efficient reaction right here. Lactic acid is a little less efficient, but large multicellular things that don't live in the water, and multicellular things in general, can't do alcohol fermentation because the alcohol concentration gets too high and it becomes biologically damaging, basically drunk, and if the alcohol level gets too high, you can die from it, alcohol toxicity. Lactic acid is far less toxic and can be metabolized in a multicellular organism. So the purpose of both of those reactions is to get rid of the products of glycolysis. So the pyruvate needs to be gotten rid of. We convert it into lactate in lactic acid fermentation. This is transferred via your blood to your liver, and your liver actually takes lactate and metabolizes it, breaks it down, making some more ATP, but somewhere else, not skeletal muscle cells. And then the rest of the reaction is all about regenerating NAD. So the NAD NAD picks up electrons in glycolysis. The NADH, if we don't have any place to give the electrons to, all of your NAD molecules become NADH, and this stops, and we can't make our 2 ATP that we're always making right here. So we need to regenerate these NAD molecules from these NADHs. So what happens in the fermentation pathway, we kill two birds with one stone, as they say. We take NADH, dump its electrons onto pyruvate in a different form to create this lower energy molecule called lactate. And now the NADHs are free to be reused for glycolysis, which generates ATP. Not a lot, but enough to keep most simple, not super exciting cells alive. And the lactate is a waste product here. It can either be dumped into the environment, or in the case of something like us, it's metabolized in another organ, the liver. Alcohol fermentation is similar, more efficient, and let's see, I don't have an animation for that one right here. I mean, I don't have a flowchart for it, but what happens in that particular case is the pyruvate is broken down into CO2 and acetaldehyde, and then acetaldehyde is given a proton and some electrons from NADH, making ethanol. That's ethyl alcohol, the stuff that's in alcoholic beverages. And the NAD is now regenerated so that it can participate in more glycolysis reactions that generate ATP for the cell to use. And the pyruvate is gotten rid of by dumping it into the environment as ethanol. Ethanol dissolves in water very effectively and leaves a single-celled organism into water really quickly and efficiently. All right, a last couple, and we'll take a look at this here at the end. I'll play this at the end. Some other things that we want to talk about. So I told you that the cellular respiration process uses organic molecules as fuel, and then we went and looked at just glycolysis, just that process using glucose as a starter. But quite a few other carbohydrates, anything really that has six carbons in it, can feed into the glycolytic pathway. All right. And anything with 3 carbons in it, including a lot of amino acids, can go in as be modified and turned into pyruvate. Some of those things that just have two carbons on them, like the fractions of fatty acids that are broken apart in fatty acid beta oxidation. Those 2 carbon fragments can be dumped into the citric acid cycle, where acetyl-CoA, a 2 carbon molecule, is attached to something else right here, and we can extract ATP that way. So the point is, we can use lots of molecules of different sizes as fuels and dump them into various steps in the process. We don't have to start with glycolysis. We can start with pyruvate, or we can start at the citric acid cycle. Wherever things can be dumped in here, we can go ahead and use them. And so this is why we can use lots and lots of different molecules as a power source for our metabolism. You can power yourself with lipids. You can power yourself with proteins. You can power yourself with carbohydrates, all kinds of different ones. The most efficient is this pathway using glucose. Here's an example of using the, and this is why we study it in introductory biology, the cellular respiration pathway, and instead kind of look at it as a way to take out or introduce things. So we can add glutamate into this process right here and run glutamate through the last steps of the citric acid cycle right here and make, I believe, one more NADH, one FAD, one GDP, and one more NAD right here. So we can ultimately make a little bit of ATP with glutamate as a fuel. That's an amino acid. But we can also take ketoglutarate, this thing right here, out of the citric acid cycle and make glutamate out of it, and use glutamate to make these amino acids. So what we're looking at right here is this system's ability to participate in us converting one amino acid into another amino acid and making a lot of the amino acids that we can make. We can't make all of them. There's 8 that we must get in our diet, but we can use those 8 to make all of these amino acids using steps and processes of the citric acid cycle, and indeed the pyruvate to acetyl-CoA oxidation process, and the glucose to pyruvate glycolytic pathway. We can use all of that as a mill to make various molecules that are useful to us. We can also make glucose. We can make pyruvate from acetyl-CoA by running these things backwards. There's lots and lots of possibilities, and all your cells do this all the time. So that's the relationship between cellular respiration and the larger, more complex metabolism going on inside your cells. It's very central. Glycolysis, oxidation of pyruvate into acetyl-CoA, introduction of acetyl-CoA, binding it to oxaloacetate to make citrate, and this then breaking that apart in a series of steps, generating free electrons, generating ATP, generating more electrons, is the citric acid cycle. Round and round it goes. Finally, this process is controlled by feedback processes. All right. So for instance, glucose is regulated very, very heavily. The process of making ATP and ADP is regulated via feedback regulation processes. And one thing we want to talk to you about is the, well, we'll go ahead and look at this right here. You don't have to memorize any of this, but various steps along the process, the electron transport chain, citric acid cycle, pyruvate to acetyl-CoA, and even glycolysis, all of them have an enzyme that is inhibited by one of the products of the reaction, all right, or stimulated by a product of the reaction. So a whole lot of ATP inhibits isocitrate dehydrogenase, stops the citric acid cycle. A heck of a lot of ADP, adenosine diphosphate, actually stimulates the citric acid cycle, makes it run faster. So same thing with these other pairs. Here's something that decreases glycolysis. Here's something that increases glycolysis. Here's something else that decreases glycolysis. Here's something that increases the oxidation of pyruvate. Here's something that decreases it, etc., 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 all the way through. And notice again and again, ATP shows up as an inhibitor. Decrease, 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 decrease. And ADP, and certainly AMP, should it show up, adenosine monophosphate, acts as a stimulant, driving more and more of this process. So the amount of ATP that you've got, and to a lesser degree, the amount of electron carriers in their reduced form, contribute to regulating of the entire cellular respiration pathway. Very nifty. And that concludes this lecture, and this entire recording is just shy of 60 minutes long.